The deepest point in the ocean sits nearly 7 miles below the surface. Nothing with bones should survive the pressure down there. A small team lowered a baited camera into the darkness anyway, pointed it at the seafloor, and left it running. And what swam into frame at that depth had no business being alive. The trench that shouldn't exist. The Mariana Trench sits in the western Pacific Ocean, east of the Mariana Islands and roughly 200 miles southwest of Guam. It stretches for more than 2,550 kilometers in a crescent shape across the ocean floor, averaging about 69 kilometers wide. None of those numbers convey quite how extreme this place is until you put them alongside something familiar. If you took Mount Everest, the highest point on the land surface of Earth at 8,849 meters tall, and placed it at the bottom of the Mariana Trench, the peak would still sit more than 1.5 miles underwater. The deepest point of the trench, a section of seafloor called the Challenger Deep, reaches approximately 10,935 meters below the surface, with a measurement uncertainty of plus or minus 6 meters based on a 2020 high-resolution bathymetric survey that produced the most precise depth figure available. That depth is not an accident of geography. It is the direct result of one of the oldest and most patient geological processes operating on this planet. The Mariana Trench formed where the Pacific Plate, the largest tectonic plate on Earth, meets and slides beneath the smaller Mariana Plate. This process, called subduction, happens because the Pacific Plate is older and denser than its neighbor. Oceanic crust at the western edge of the Pacific Plate is up to 170 million years old, according to Robert Stern, a geophysicist at the University of Texas, Dallas, making it some of the oldest seafloor anywhere on the planet. That age means it has cooled and contracted significantly, giving it a higher density and a tendency to sink when it encounters lighter, younger rock. At the rate of roughly a few centimeters per year, the Pacific Plate bends and dives beneath the Mariana Plate and descends into the Earth's interior. That bending creates the crease in the seafloor that forms the trench itself. The subducting rock melts as it descends into the hot mantle, and the resulting magma rises back toward the surface further to the west, which is what created the chain of Mariana Islands visible on any map of the region. At the trench itself, the pressure at the very bottom of the Challenger Deep reaches 1,086 bar, or approximately 15,750 pounds per square inch, over 1,000 times the pressure at sea level. At that depth, temperature sits between 1 and 4 degrees Celsius year-round, just above freezing regardless of the fact that the trench lies in a tropical stretch of the western Pacific. No sunlight penetrates below roughly 200 meters in ocean water. From 200 meters to nearly 11,000 meters, the Challenger Deep operates in absolute permanent darkness, broken only by whatever artificial light a submersible or camera lander brings with it. These are the conditions that anyone, or anything, going into the Mariana Trench has to contend with. The first 2 people who went there. For most of human history, the Challenger Deep was simply a measurement on a chart. The British Royal Navy research vessel HMS Challenger first recorded a depth of 8,184 meters there in March 1875. A second HMS Challenger expedition in 1951 returned with better equipment and confirmed a sounding of approximately 10,900 meters, giving the feature the name it still carries. Actually reaching the bottom required technology that did not exist for most of the 20th century. The bathyscaphe Trieste, designed by Swiss physicist Auguste Piccard and later sold to the US Navy, was the first vessel capable of carrying humans to Hadal depths. On January 23, 1960, Jacques Piccard, son of the designer, and US Navy Lieutenant Don Walsh climbed into its steel pressure sphere, just 2 meters in diameter, suspended beneath a massive buoyancy tank holding roughly 130,000 liters of gasoline. The descent took 4 hours and 47 minutes. At 9,000 meters, the outer window pane cracked loud enough to shake the entire vessel. They confirmed no pressure drop and kept going. At 1:06 in the afternoon, the Trieste settled onto the seafloor at approximately 10,912 meters, onto a pale brownish ooze of microscopic algae. They stayed 20 minutes. Picard reported seeing what looked like a flatfish resting on the sediment before swimming away. Later, researchers questioned it, suggesting he may have seen a sea cucumber in the disturbed silt. Whatever it was, something was alive down there. Walsh and Picard returned to the surface, expecting others to follow within years. More than half a century passed instead. 52 years of silence, the U.S. Navy scaled back its deep submersible program after the Trieste mission, and no human returned to the Challenger Deep for over 50 years. Robotic vehicles filled the gap. The Japanese ROV Kaiko reached the bottom in 1995 and 1996, collecting sediment samples, and the Woods Hole Oceanographic Institution's ROV Nereus reached 10,902 meters in 2009. The first human to go back was filmmaker James Cameron, who descended alone on March 26, 2012, in a 7-meter submersible called the Deep Sea Challenger that had taken 7 years and roughly $10 million to build. He reached approximately 10,908 to 10,912 meters, the first solo crewed dive and the first human visit since 1960. Cameron had high-definition cameras recording everything. What they captured was close to nothing. He saw skid marks left by the Kaiko more than a decade earlier, still perfectly visible in the undisturbed silt. Small sea cucumbers, single-celled organisms. He described the overall impression as one of complete isolation and called it the loneliest place on Earth. His expedition did recover scientifically useful samples, including evidence of never-before-seen microbial communities in the nearby Serena Deep, but the visual record was stark. The deepest place on Earth, seen through a camera for the first time, looked like a featureless pale desert with no visible end. The expedition that changed everything. In 2018, Victor Vescovo, a retired U.S. Navy Reserve officer, launched the 5 Deeps expedition with the goal of reaching the deepest point in each of Earth's 5 oceans. His vehicle was the DSV Limiting Factor, the world's only commercially certified submersible capable of reaching any ocean depth. The Mariana Trench leg took place in April and May 2019. Vescovo made multiple dives, reaching 10,928 meters on his deepest descent. 3 robotic landers deployed alongside him sat on the seafloor for hours, with cameras running continuously. The expedition collected over 500 hours of high-definition footage across 13 locations and discovered what appeared to be 3 new species, including an amphipod found at the very bottom of the Challenger Deep. Vescovo spent approximately 4 hours on the seafloor, longer than any previous visitor. He described it as peaceful and surreal. He also described finding a plastic bag and candy wrappers on the sediment, telling reporters it was very disappointing to see obvious human contamination of the deepest point in the ocean. In June 2020, Vescovo returned with NASA astronaut Dr. Kathy Sullivan, the first American woman to walk in space. On June 7th, Sullivan became the first woman to reach the Challenger Deep, and the first person in history to have visited both outer space and the deepest point in the ocean. After surfacing, the two called the crew of the International Space Station orbiting 254 miles above Earth. Sullivan described the seafloor as resembling the moonscape she had seen from orbit, pale and still and impossibly remote. What the cameras actually caught. The most significant footage from the Mariana Trench came not from crude submersibles, but from simple baited lander systems. Marine biologist Alan Jameson has spent more than 15 years deploying these weighted, self-contained frames into Hadal trenches. They sink to the seafloor, sit for hours with cameras running, and record whatever approaches the bait in the surrounding darkness. In 2014, a lander deployed from the Schmidt Ocean Institute's research vessel Falkor filmed a completely unknown snailfish species at 8,143 meters. The white, translucent creature had broad, wing-like fins and an eel-like tail. Jameson said it looked like nothing his team had ever seen and matched nothing in the existing scientific literature. It was never collected, only filmed, and researchers gave it the informal name the ethereal snailfish. On May 18, 2017, a lander operated by the Japan Agency for Marine Earth Science and Technology filmed a formally named species, Pseudoleparus swirri, at 8,178 meters. The footage showed a pale, scaleless creature roughly 20 to 30 centimeters long, swimming calmly through conditions that would kill almost any other vertebrate on Earth within seconds. In August 2022, a team led by Jameson filmed an unidentified juvenile snailfish at 8,336 meters in the Izu Ogasawara Trench, south of Japan, the deepest fish footage ever recorded. The Natural History Museum London's Rupert Collins noted that between 8,200 and 8,400 meters, fish can no longer accumulate sufficient TMAO to keep their proteins functioning, and that this biochemical limit appeared to hold across every measurement collected so far. The chemistry that sets the limit. The reason any fish can survive at these depths at all comes down to a molecule most people have never heard of. Ordinary proteins, the building blocks that run every cellular process in every living organism, begin to lose their correct shape under sufficient physical pressure. This unfolding, called denaturation, disrupts protein function and kills cells. At depths approaching and exceeding 8,000 meters, the pressure should be more than sufficient to cause this in any normal vertebrate animal. Snailfish, and other deep-sea animals, survive by accumulating a chemical called trimethylamine oxide, commonly abbreviated as TMAO. TMAO belongs to a group of pressure-counteracting molecules scientists call piezolites. These compounds work by stabilizing the three-dimensional shape of proteins even as external pressure pushes against them, effectively acting as a molecular scaffold that holds the protein's structure in place against forces that would otherwise deform it. Biochemist Paul Yancey of Whitman College, along with research colleagues, noticed something striking about TMAO levels in fish collected from progressively deeper environments. The deeper a fish typically lives, the more TMAO its tissue contains. This correlation was consistent enough that Yancey's team worked out a theoretical prediction from it, published in 2014. If TMAO concentration scales linearly with depth, and if there is an upper limit to how much TMAO a fish's cells can hold before the TMAO itself starts disrupting cellular processes through osmotic effects, then there is a hard biochemical ceiling on how deep any bony fish can live. Their calculation placed that ceiling in a range of approximately 8,200 to 8,400 meters. Jameson, when asked whether he was surprised to find a snailfish so close to the theoretical limit, noted that the 8,200 meter ceiling had always come with the caveat that temperature plays a role alongside pressure, since both have similar destabilizing effects on proteins. The trenches near Japan run slightly warmer than the Mariana Trench, and 2 years of research had already pointed to this region as the most likely place to find the deepest fish on Earth. If the kind of science that leads researchers to predict exactly where a record will be broken before anyone goes looking is what keeps you watching, subscribing is how you make sure the next discovery reaches you. Jameson described the find not as a surprise, but as a confirmation of exactly what the research had predicted, which is its own kind of remarkable. The fish filmed at 8,336 meters represents, by the best current understanding of Hadal biology, something very close to the maximum depth at which any fish on Earth can possibly operate. The wall is not made of rock or steel, it is made of chemistry. Who else lives down there? Fish are only one category of animal that cameras have documented in the Hadal zone of the Mariana Trench. The life that cameras have captured there is far stranger and more varied than the empty moonscape James Cameron described from his view through a small porthole. Sharing the deepest zones of the Mariana Trench with the snailfish are supergiant amphipods, scientifically called Alicella gigantea. Typical amphipods, the shrimp-like scavengers found throughout the world's oceans, grow to about a centimeter in length. Alicella gigantea grows past 30 centimeters, making it 20 to 30 times larger than its shallow water relatives, and has been documented at depths ranging from roughly 3,890 meters to nearly 8,931 meters, deeper than any fish has been recorded at the Mariana Trench. The reason for this dramatic size increase is not fully understood. The leading theory, called deep-sea gigantism, proposes that the combination of crushing cold and extreme pressure slows metabolism enough to allow cells to keep growing for much longer than they would in warmer, shallower water, producing animals that live considerably longer and grow considerably larger than their relatives elsewhere. Genetic analysis of Alicella gigantea populations across multiple trenches, including the Mariana, suggests that a single connected breeding population may span more than half the world's oceans, with nearly identical DNA showing up in trenches thousands of kilometers apart. The strangest resident of the deepest Mariana Trench zones is something that barely qualifies as an animal in any familiar sense. Xenophyophores are single-celled organisms, technically protists, that grow to approximately 10 centimeters in diameter, making them among the largest single cells known to science. A single xenophyophore is one cell, with one nucleus, doing everything a cell does, but at the scale of a large fruit. They live on the seafloor, constructing irregular structures from sand grains, sponge fragments, and shell material accumulated from the surrounding sediment. These structures provide microhabitats for smaller organisms, including bacteria, tiny crustaceans, and other meiofauna, making xenophyophores a kind of living apartment building for the Hadal zone's smallest residents. In July 2011, a Scripps Institution of Oceanography expedition deployed camera landers into the Sirena Deep section of the Mariana Trench and documented xenophyophores at a depth of 10,641 meters, shattering the previous depth record for the species by nearly 2 miles in a single expedition. Scripps marine microbiologist Doug Bartlett, who organized the expedition, said at the time that the identification of these giant single cells in one of the deepest marine environments on the planet opened up a whole new habitat for further study. Research has also found that xenophyophores concentrate heavy metals from the surrounding water at unusually high levels, including lead, uranium, and mercury, suggesting they play a role in the deep-sea biogeochemical cycling of elements that would otherwise remain dispersed in the sediment. Sea cucumbers move across the seafloor in considerable numbers at Hadal depths, described by multiple expedition reports as among the most abundant visible animals at certain depths within the trench. Stalked crinoids, which look like underwater plants but are animals related to starfish, anchor themselves to hard surfaces. Polycate worms tunnel through the soft sediment. The bacterial and archaeal communities living in the trench sediment represent an entirely separate dimension of life that camera footage barely captures, requiring physical samples for analysis, and which ongoing research suggests may represent some of the most ancient and biochemically unusual microbial lineages on Earth. The disturbing thing sitting next to the fish. Among everything cameras and sampling equipment have found in the Mariana Trench, one category of discovery stands apart from the rest and demands its own section. In 2017, the same year the Mariana snailfish was filmed at its record-breaking depth, a research team led by Alan Jameson at Newcastle University published a study in the journal Nature, Ecology, and Evolution examining tissue samples from small amphipods recovered from 6 deep-sea trenches, including the Mariana, using landers of the same type used to film the snailfish. The results of chemical analysis of these samples were, in Jameson's own description, shocking. Every single amphipod tested, from every trench examined, contained polychlorinated biphenyls, known as PCBS, and polybrominated diphenyl ethers, flame retardant chemicals in their fatty tissue. PCBs were banned in most countries by the late 1970s due to their links with cancer and other health effects. The contamination levels were not trace amounts. The most polluted amphipod samples recovered from the Mariana Trench carried PCB concentrations roughly 50 times higher than crabs sampled from the Liaohe River in China, one of the most industrially polluted rivers in the world. Jameson told The Guardian afterward that the team had always assumed the deep ocean was pristine and remote, and that the results showed that assumption was completely wrong. This pollution did not drift down gradually. Scientists believe PCBs and similar persistent organic pollutants reach the Hadal zone primarily through marine snow, the slow rain of organic matter that sinks from the surface ocean. Plastic fragments, dead organisms, and other material carrying these chemicals absorb into fatty organic material as they sink, and that material is eventually eaten by scavengers like amphipods at the bottom of the trench. Once a chemical reaches the sediment of the Challenger Deep, there is essentially nowhere else for it to go. It accumulates, decade after decade, in a place that is almost never sampled and almost never cleaned. The plastic bag Victor Vescovo observed during his 2019 dive sites in that same context. A study published in 2018, using data from the Deep Sea Debris Database, a collection of images and video from 5,010 dives over 30 years, documented what was then the deepest known piece of plastic, a shopping bag found at approximately 10,975 meters inside the Mariana Trench. Researchers analyzing water samples from the southern Mariana Trench, published in the journal Geochemical Perspectives Letters in 2018, found microplastic concentrations of up to 2,000 pieces per liter in the most contaminated trench sediments, and 11.43 pieces per liter in water samples at 10,903 meters depth, 4 times higher than surface waters in the open Pacific Ocean. Newcastle University stated in their press release accompanying Jameson's study that it was highly likely that there were no marine ecosystems left that were not impacted by plastic pollution. The Hadal zone, they wrote, was likely one of the largest sinks for microplastic debris on Earth. What this means for how we understand life. The combined picture that camera footage, lander samples, and crude submersible dives have assembled of the Mariana Trench over the past decade upends almost every assumption that was common earlier in the 20th century. The assumption that the deepest ocean was essentially lifeless persisted well into modern research. As recently as 2014, baited camera landers had been sent to regions deeper than 6,000 meters in 5 Pacific trenches 14 times without ever photographing a fish. That specific statistic comes from researchers at National Geographic who were documenting the state of Hadal fish knowledge at the time the Mariana snailfish was first formally described. The Hadal zone had been described in textbooks as too extreme, too isolated, and too food poor to support vertebrate animals. The snailfish and their relatives proved that description wrong. The assumption that the deepest ocean was pristine and untouched by human activity was similarly widespread. Oceanographers routinely described the deep ocean as a refuge from anthropogenic impact, a last reserve of uncontaminated biology. Jameson's PCB study and the subsequent cascade of research on microplastics in Hadal sediments demolished that assumption entirely. What has replaced these older assumptions is a more complicated picture. The Mariana Trench is not empty, but it is also not what popular imagination tends to project onto it. It is not inhabited by enormous, fearsome creatures. The genuinely large animals of the ocean, giant squid, sperm whales, and similar megafauna, do not reach Hadal depths. Giant squid are known to dive to roughly 1,000 meters. Sperm whales, the deepest diving air-breathing animal on Earth, have been recorded at depths up to about 2,250 meters during extreme feeding dives, barely a fifth of the way to the Challenger Deep. The genuine residents of the Hadal zone are soft, small, chemically specialized animals built to manage pressure at the molecular level rather than overpower it through size. The trench is not pristine, but it is also not uniformly contaminated. Active microbial communities carry out biochemical processes at the very bottom of the Challenger Deep that researchers are still working to fully characterize. Some of these communities may operate through chemical energy rather than photosynthesis, making them independent of sunlight in ways that have drawn serious scientific interest regarding questions about where life can exist and how it first originated. Verdict. Here is where things actually stand. The Mariana Trench stretches 2,550 kilometers across the western Pacific Ocean floor, reaching its deepest confirmed point at the Challenger Deep at 10,935 meters, established by a 2020 high-resolution survey with an uncertainty of plus or minus 6 meters. The first humans to visit this depth were Swiss oceanographer Jacques Piccard and U.S. Navy Lieutenant Don Walsh, who descended in the bathyscaphe Trieste on January 23, 1960, and stayed for roughly 20 minutes at approximately 10,912 meters before returning to the surface. No human returned for 52 years. James Cameron made the first solo crewed dive in March 2012 aboard the Deep-Sea Challenger, reaching approximately 10,908 to 10,912 meters. Victor Vescovo reached 10,928 meters in April 2019 as part of the Five Deeps expedition, deploying landers that recorded over 500 hours of high-definition footage and discovering what appeared to be 3 new species. Dr. Kathy Sullivan became the first woman to reach the Challenger Deep and the first human to have visited both outer space and the ocean's deepest point on June 7, 2020. Cameras deployed inside the Mariana Trench have filmed a pseudo-Liparus snailfish at 8,178 meters in 2017, close to the biochemical depth limit of 8,200 to 8,400 meters predicted by biochemist Paul Yancey based on TMAO concentration measurements. A separate related species was filmed at 8,336 meters in the Aizu Ogasawara Trench in 2022. Currently the deepest fish footage in existence. The Serena Deep section of the Mariana Trench hosts xenophyophores, single-celled organisms approaching 10 centimeters in diameter, recorded at depths of 10,641 meters by a 2011 Scripps Institution of Oceanography expedition. Supergiant amphipods of the species Alicella gigantea, growing to over 30 centimeters, have been documented at depths approaching 8,931 meters. A 2017 study published in Nature Ecology and Evolution by Alan Jamieson's team at Newcastle University found PCB contamination in amphipods from the Mariana Trench at levels approximately 50 times higher than crabs from heavily polluted Chinese rivers. Research published in 2018 documented microplastic concentrations of up to 2,000 pieces per liter in Mariana Trench sediments. A plastic shopping bag represents the deepest confirmed piece of human-made debris, documented at approximately 10,975 meters. The deepest place on Earth is not empty. It is also not untouched. What cameras found at the bottom of the Mariana Trench was stranger, more delicate, and more compromised than almost anyone predicted before anyone actually went there to look. If this is the kind of story that keeps you watching, subscribing means the next one reaches you when it comes out.